Thank you so much, everybody, for joining our event. Whosoever is watching us on YouTube Live on Sunday morning, 10:30. And my first of all greetings to Dr. Vinay K. Nandicoori, sir, who serves as the current director at CCMB Hyderabad. Thank you so much, sir, accepting for invitation and talking to the students today. Sir, you are on mute.
You're welcome and it's a pleasure.
I request Bhakti Sadguru to please introduce Dr. Vinay Nandicoori, sir, for today's event.
Good morning, everyone. Let me quickly introduce our today's speaker, Dr. Vinay Kumar Nandicoori. He's a distinguished Indian immunologist and biotechnologist celebrated for his pioneering research on Mycobacterium tuberculosis, the causative agent of tuberculosis. He currently serves as the director of the Center for Cellular and Molecular Biology in Hyderabad, India, where he contributes to lead innovative research in the field of infectious diseases. Dr. Nandicoori's academic journey began with a master's degree in biotechnology from the Indian Institute of Technology, Bombay. He earned his PhD in molecular and cellular biology from the Indian Institute of Science, Bangalore. Subsequently, he undertook postdoctoral research at Texas A&M University and the University of Virginia, where he specialized in cellular signaling mechanisms. Before his current role at CCMB, Dr. Nandicoori served as a staff scientist at the National Institute of Immunology, New Delhi. During his tenure at NII, he made groundbreaking contributions to the understanding of kinase-mediated signaling towards in understanding of kinase-mediated signaling networks in Mycobacterium tuberculosis. His research focused on eukaryotic like serine threonine protein kinases and their critical roles in regulating various cellular processes such as cell division, cell wall synthesis, transcription, secretion, and the pathogen survival strategies within the host. Under his leadership at CCMB, Dr. Nandicoori continues to push the boundaries of tuberculosis research, aiming to identify novel therapeutic targets to combat the disease, especially against multi-drug resistant and extensively drug-resistant strains. Dr. Nandicoori's contributions to biosciences have been widely recognized, earning him prestigious accolades such as the National Bioscience Award for Career Development from the Department of Biotechnology, Government of India in 2010, and the NASI Scopus Young Scientist Award in 2009. In addition to his administrative and research roles, Dr. Nandicoori has an extensive publication record, shedding light on the molecular mechanisms that enable Mycobacterium tuberculosis to persist within hosts. His work not only advances scientific understanding but also offers hope for innovative solutions to tackle tuberculosis, a persistent global health challenge. So we are very honored to have you today, and I'm looking forward for the event. Thank you so much.
Thank you. Thank you for an extensive introduction.
Sir, we are greatly honored today to have you on SciComm Synapse. Without passing much time today, we would directly start the event. And the first thing that I would like to ask you is that when you were pursuing your PG in biotechnology from IIT Bombay, so what inspired you to do a PhD, or maybe before that time that like I would really go into this field?
I actually was not pursuing biotechnology or biology till about BSc. I was actually in chemistry, physics, and mathematics. So Bombay in those days was the only place in India where you could write a chemistry entrance and get into biotechnology. And in fact, that's how I got into it. At the initial, initially when I was in IIT, I really didn't think far beyond just surviving and trying to do better. I mean, I tried to do my best, I would say that much. So after 2 semesters, I had an opportunity to do summer training at BARC in Bombay. There I was working in a laboratory, and there I had to work on removal of lactose from milk using yeast actually. So that is the time I kind of figured that I actually have a pretty good experimental skill sets. So like I can actually understand how experiments are designed, executed, and reasonably well executed. So that's kind of gave me a feeling that I can actually do this and I like it. And when I came back, I had a master's project, which was on, it's like a enzyme called arginase. And the lab, basically, we were doing our master's and in parallel pursuing the project. And this was a work on Aspergillus niger. So all this time, I kind of realized that this is something that I liked. So after master's, it was not realistically a something that I had to think about. That I wanted to pursue PhD was obvious to me. Question was where, and I ultimately ended up in University of Sciences, Bangalore. That's
your journey, how you reached to PhD, sir. So, since you joined Professor Umesh sir's lab till I am sure, like transition from your masters to joining the PhD, how was your experience when you started your PhD? Was it the same way that you thought about it, or was it was a bit different?
PhD is something that you basically, when I started out, that I started out in 1991 August, I was the first student of Professor Washington. So we actually had an empty lab. And one thing that I had was my boss's time, lots of it. So, and he's a very good teacher. So that basically kindled a lot of experimental biology interest, or continued that. And initially, I wasn't thinking about publishing papers or really this thing. I always think in short terms. I look at it as what am I going to learn, right? I focused on experiments. I kind of tried to learn everything that has been taught to me to the best of my ability and reproduce it as much as flawlessly as possible. That was the effort. And those days, molecular biology was different. Management, column preps were not really the norm. People had to do sequencing classical way, means running those long gels, and radioactivity was part and parcel of everything that we do, including assays and other things. So there was a lot more technical skills involved in those days compared to today, relatively. I mean, I'm not saying it doesn't require today, it does require. And initial first year was simply learning. Second year was executing some of those things. I was lucky enough to get a publication in my third year because my boss actually, that is Professor Washington, came with a project. He gave it to me and we worked on together, and mostly I worked and he gave me the ideas. But after the publication is where I really science started, because when you get a publication so soon in your career, you tend to think that you have achieved something. And then the reality actually sinks in that it's only a publication, it's nothing more than a bus stop in a long journey. So yeah, you go through these downs and you go through these ups and you go through all kinds of outside roller coaster, and that's no different for me than I'm sure it is quite a lot of people who are currently pursuing PhD are facing. 5 years, 4, 5, 6 years is a long haul. It is very difficult to keep our enthusiasm at that level and consistently so. You will have your bad days, you will have your fantastic days, you would have routine days. So it's the idea is when you have bad days, it is important to have friends with whom you can hang out, or some way to kind of come out of those bad days and effectively start the next day as a fresh one. And that's important for if you really want to pursue this and come out unscathed from the PhD experience, because you're learning initially and then you don't get the results that you think you want, and then you start feeling that you're so unlucky that you're not getting those results. But it's part of the journey. It's like you have a hypothesis. Out of 5 hypotheses, 1 works, 4 don't. But the fact is you have to face those 4 also and come up with an alternate hypothesis. Requires a lot of tenacity and requires a lot of mental strength. And over a period of time, we become better at it. It takes a while. So I would say first 2 to 3 years are a breeze. You're happy being there. You're happy learning. You're happy with everything that happens around you. And after that, the reality slowly sinks in that, there is a lot to be done, and the career is a long haul. You have a PhD, and then you have a postdoc, and even when you become faculty, it doesn't end. It's like it's a really a long haul, and it requires a lot of motivation, a lot of strength, a lot of focus.
Absolutely, sir. I think the days when you did your PhD, 1991, 97, yeah, the science was very different than what we do today in the lab
Correct.
today. Agreed. Sir, so like, can you share your any memorable experience of an experiment, if you remember right now that like maybe like one experiment that you would remember your entire life that yeah, I think this was the experiment which in my PhD which you would remember.
Yeah, I do actually. Quite a lot of us do. I believe that I remember most of my failures than the success. So that's
Uh-huh. Uh-huh.
one. To start with, we had to do manual sequencing. Manual sequencing in the sense that you start with radioactive. There are multiple steps. It is roughly 2.5 days
Mhm.
job or 2 days job, end to end.
Mhm.
So
Mhm.
I did this manual sequencing with another senior of ours in another lab. So we two did this experiment together. After 2 days, when ultimately we looked at the autoradiogram, which is the image that you see after running a sequencing gel, getting it and exposing it to the thing, exposing it to in an X-ray cassette, you get an autoradiogram. And when we looked at the autoradiogram after 2 odd days, we realized we failed, and badly so, right? And then we kind of figured that maybe the way to, like some step somewhere, something has gone wrong. You have to repeat it, right? I mean, you can't let that go. So that was almost at night, 10 o'clock, I think we developed this autoradiogram. And we were very disappointed, very upset, went out for a chai, both of us, came back, and I said, okay, let's start again. And we started again. We didn't give up on it. We started again. We finished that in the next 1.5 days again. We put the autoradiogram, and this time the thing has worked. So we went from absolute despair to complete elated feeling just about 2 days later. Means we experienced end-to-end emotions, one experiences with failed experiments to success in just a matter of 2 days. And that stayed in my mind that you just can never give up.
That's true, I think. And I mean equally the contribution of PhD student and also the professor who is guiding you when there's a new lab makes I think also a difference at that point.
Yeah, having a right mentor helps, because if you have a mentor who's very patient and wants all the time data, and that puts a lot of stress on already stressed life. So, but however, if you are given a bit of a room to maneuver and kind of figure it out yourselves, it certainly helps to have that particular worry taken off, and then you can focus on what is in your hand and what is actually something that you can control, or at least try to control. So yeah, it does help to have a good mentors, and I mean, unfortunately, the life is such that you'll have mentors of every kind.
Yes, sir, definitely. Sir, so since you mentioned about the biochemistry and the molecular biology that you used to do all the sequencing, so when did you thought of that? Yeah, I think I want to finish my PhD and then go for a postdoc. And did you think that would you go to US or?
It was this is, I would say this way: almost every one of my seniors was in US. I was in
Mm-hmm,
Europe, and that was the norm. And so it
mm-hmm,
was
mm-hmm,
almost like a path that is already laid out. You just have to follow it. And the question
mm-hmm,
of when you end up generating decent enough CV, then you'll start thinking of it. I did it when I
mm-hmm,
finished my PhD in 1997. I submitted my thesis. Before that, I
mm-hmm,
started applying. But I would say that one has to do this particular thought process at least a year earlier than that, or at least six
mm-hmm,
months
mm-hmm.
earlier than where I started. And so that you can actually make your path going forward much clearer, much easier. The opportunities now to then are different. At least by the time our time came, we already had email. Prior to that, it was literally writing letters to your future postdoc mentors and it is writing a letter and putting it in a post box and hoping that you will get a response. And so it's different, and it is as the time has progressed. Now people go for international conferences, and there they
Yeah.
interact, they try to fix their, interact with people, set up interviews, go to the labs. Things have changed. Things have changed in a very big way. I mean, it was different, but I was more or less clear that I would be pursuing a postdoc minimally. What I was not, however, clear is whether I want to stay in academics or whether I want to get into industry, whether I want to be in, contributing in getting into an industry for later part of my life as a career, or whether I want to continue and become a faculty. These were the only two paths that were ahead of us when we were finishing PhD. Now there are multiple other paths. I know that, for example, you can get into IP and you can get into editorial boards. You can do many other things too. But at that point of time, I was only thinking of these two aspects. So that I wasn't clear about, but I was clear about going abroad.
Mm, that's fantastic, sir. So, like, when you finished your thesis and you left India and you reached to US and started your postdoc. So, what was the difference in the experience over there, leaving Professor Varshney's lab and then joining
See, my
a new lab?
Mr. Washington is a fantastic mentor. And I had a very, very good relation with him as a PhD mentor and a student. We had a very good relation. So it was, I mean, I was very comfortable in the lab, and the lab was highly populated. It had around 10 people odd, and I was the senior most in the lab, and there was a certain vibrant environment there. You would think that when you go abroad, you're going to get that particular vibrant environment because your whole perception is very different. We are in India, we have a vibrant environment, and when you go there, you would expect that to happen. Not realistically so. Landed in US, and the environment was very different. And I was very comfortable in the lab. So of vibrancy in terms of scientific discussions. There is a certain level of aggressiveness in science and all those things. There I started, I did quite all right there, quite well. So, over a period of time, I developed looking at science differently. I started looking at a broader picture of science. I started thinking about, how to solve problems was not difficult for me. Not anymore in the experiments, but as a problem space, right? And so that kind of started getting the feeling that I'll be quite all right in an academic setting. I mean, most probably I have what it takes to be a PI. That was basically because people are going to depend on you, right? So then I had two options. Again, I was looking at industry and academia both in parallel. In fact, I interviewed at both Reddy's and Ranbaxy when I came to India, in addition to 4 other academic places. So I interviewed in 6 places, made a specific trip for that, and actually gave talks, interacted with people, and eventually, chose academics over industry and joined National Institute of Immunology in 2004. Did I know I want to go back to India? That's something I was reasonably sure of. But that is because of that, I never even applied. Not me or my wife. Both of us were very sure of that. My wife is also a scientist. That, we did not apply for even green card. And which was very, very easy to get it in 2000s. So, we didn't apply for anything. We simply were on H1 and we applied to India, came back. Our son was around 4 to 5 years old. We thought that was the right time to move. So just moved back. So it's about thought process again. And in this thought process, it's not one person, but 2 people are
involved. So it's fantastic. So, since you returned back from US, having an experience from abroad and also at the same time doing a PhD in India, can you describe your experience when you started your own research group in India for the first time at NII?
One of the good things about being in places like NII, or for that matter CCMB, is almost everybody comes with similar background. Either they've done a postdoc in Europe, or they've postdoc in case of CCMB. Right now, we have few people who have done postdoc in NCBS here and there. But when they come to India, when they come start working, you find people with similar kind of backgrounds. So the kind of problems. You can go to some of these people, some of the senior lot, and have conversations, because it's a different ball game. You never ran a research program. You are part of a research program, right? And you never had to worry about funding. Somebody else got the funding for you. You never really had to worry about getting a reagent. Somebody else ordered it for you, right? When you are in India, you do everything end to end. You're not anymore a scientist, but you are a lead scientist or a group head or a scientist running a lab. So you need to know how to write indents. You need to know how to figure out the order process, and you need to figure out the time frames. You need to figure out how to take students and attract them to your lab, and how to manage them. And it's a different experience. I would say that's why every stage, actually, there is a learning period. If you are actually frank to yourselves, say that, okay, if I am going to go to the next step, is there going to be downtime? Yes. Would you have to really make an effort to learn? Yes. So yes, there is a learning time. And that's how I kind of believe that 6 months to 1 year again, as a fresh faculty, I had to literally learn the ropes of running a lab and mentoring students, which is a challenge.
Completely agree, sir. Sir, so can you describe what were the initial challenges when you had to establish and lab and finally publish first research article from as a corresponding author?
Challenges are manifold. I was actually thinking of a project, and once I thought about a particular project, when I started initiating the project, I wrote for a grant, I got it, and for project collapsed. It didn't work, right? I mean, whatever I thought, after 1.5 years, 2 years, I had nothing to show. And unlike many, I actually moved fields. I didn't carry much of postdoctoral work into my PA, being a faculty. Like I used to work on prostate cancer, I used to work on ERK2, and I was looking at substrates of ERK2. Leaving the fact that I wanted to look at the MAP kinases or serine kinases and look at the downstream targets, there's nothing similar to what I was doing and what I started. What I started was working on mycobacterial serine kinases. They are eukaryotic like serine kinases, but in Mycobacterium tuberculosis, a bacterial system. I had certain ideas which are coming from my eukaryotic background, and I applied them to the same kinases, right? Serine kinases. So it collapsed. So realistically, I was trying to, my idea, the whole hypothesis simply didn't work. So I had to reinvent, rediscover, and of course, along with the students. Remember, you have to keep their motivation high. You have to at least keep them motivated, and you can't say, "Oh, everything that I thought's collapsed. Now let's, I don't know what to do." You will have to have that. They need to have that confidence in you. So the journey is along with the students. So that's the challenging part of it. Keep them motivated, and when you are not realistically very happy about where you are, right? And so that there are people who are still enthused by what you have to say and work on those experiments. So it's a learning experience. Everything that you think doesn't work. There are challenges which range from end to end, like funding, getting funding, and then getting material into the lab, and then getting good students to work with you, and keeping them motivated, and so that they don't hate you by the time they leave, and then finally be productive. It's different set of challenges, and it is stressful in its own way. But again, you can't simply take your stress out on the people who work for you. You do that, your lab completely collapses. So you got to have that ability to take it in, and then kind of be still that person whom other people respect and look up to.
I think that made you a successful PI and currently the director at CCMB as well. Sir, so like since you started your research group there, you established and you mentioned that about your first project and everything. Like, how was the feeling when you became the director of CCMB, or did you already like when it was decided that you are elected as director? So when you had to move your lab and everything now to the new position.
See, none of these things happen automatically. Okay, none of these things happen without your consent. So you are
Correct.
involved on it. You know that you are actually, somebody nominates you, but they only nominate you if you want to. Nobody nominates a person who doesn't want to become a director, right? So, and then you still have to have an interaction with the committee where you have to present your ideas about the institute. So it's not like a surprise thing that happens, right? It is something that you're actually working along with somebody. People believe in you that you can be a leader, and some committee believes that you can be leading an institute and you're not going to take it down. So you understand. So these things are not completely unprepared. You actually are prepared in your mind that you're taking up a job which is a different one and a leadership position. Whether you want to, how do you transition to thinking that you can be a leader is a different matter. Over a period of time, what happens is all of us sit in various committees. Committees for equipment, committees for this, and committees for consumables, equipments, academic, admissions, you name it, various committees. Over a period of time, I got this feeling that I have decent enough administrative skills. I'm not saying scientific skills. Scientific skills have no bearing on administration. Administrative skills are different. The way you look at things, there are a set of rules that are defined. It doesn't change very much for Germany or, for that matter, any other place in India. Everywhere there are a set of rules. Some set of rules in India are more rigid. Some things are more easy to get things done. But anyway, you have set of things and set of boundaries. And how you get your things done, that is determined when you are faculty itself. And if you're capable of doing that, then you know that you have what it takes to actually run something. It's an assumption. All these things are your thought process, and many other people over a period of time develop that feeling that okay, he can be a good director. He may be able to run a place effectively. You understand? So it's kind of feedback, positive feedback, feed forward loop that kind of feels that okay, you can manage it. And ultimately, once you get the job, initially you are very happy that you are going to be the director of a place, but I was actually very, very scared too, because it's a new job, it's a new setting, and I'm an outsider going into CCMB. I had to actually, first of all, understand the place, and then I have to run it effectively, and I have to know every person in that organization, and and I have to become part of the organization. This outsider tag can be pretty very difficult to wear on your sleeves. You understand? So
Completely.
I would say it's a different sort of experience, but again, 6 months to 1 year, the way I look at everything in life is you change the job, you change what you are doing, you're to learn. A continuous
So
learning experience.
I think that's something that is very valuable from you today, sir. Continuous learning experience. Sir, so like since you became the director of CCMB, now how do you really balance the administrative work and
Difficult.
the
Very difficult. Not difficult, very difficult. Highly demanding. The job in itself, you're there as a director, means you have a responsibility to the institute, right? So what I do is I mean I'm not exactly a workaholic or anything, but I start my day at around 8:15ish. I start my days with interacting with students most of the time, and after that I go and get to my administrative hat, and I do that till almost 6:30 in the evening. So I sit around 9:30 till around 6:30, and realistically hardly major breaks nowadays, because even my lunch is almost a working lunch. So so after that I get back to the lab part of it, and I talk to them. What I do, one thing that I have over a period of time learned, is that on Saturdays I only do as much as possible. There are times it is not possible despite that. At least 2 Saturdays out of 4, I only focus on lab. At least 2 out of 4, sometimes 3. There are good months where all 4. I'm making an active effort to understand what kind of students are there, what kind of problems they are facing, and how to solve them. So that's the only way I can actually keep it going. And then students, whenever they send me a message, they ask something of me. I don't say that I'm busy. I believe I have a responsibility to them. So I do my best. Even they understand that sometimes they come at 8:30 in the morning, and they come without complaining. They also understand my constraints, right? So I've been very lucky to have talented and self-motivated students. That also helps.
Sir, that's
What being
fantastic.
successful at this level as a faculty and anything else depends on what kind of students or what kind of people work for you, right? If they are self-motivated, you have won the battle. Half the battle is won. The rest of it is only to kind of keep it going. And I was always lucky to have talented students, fantastic talented students.
Absolutely, sir. Absolutely. Sir, so like since you are a PI now and looking at your PhD journey and also maybe your favorite PhD students or few PhD students who would have published really good for you as well. I hope everyone would have done that. But what tips would you like to give to PhD students who are pursuing their PhD right now, like me? That
Because
okay.
I think more or less what I said when I was a student, whatever I learned is tomorrow is another day, right? I mean, so let's not worry about becoming. If I had a bad day, I always feel that next day should be started afresh. You cannot carry over today's bad experiment, move into the next one. Tomorrow, I mean, that when you start the next day, you should look at it without really getting worked up. And I kind of implement that policy for everything. I had bad days as a director. I will have bad days because I receive a bad review from a journal. You will have bad days. But in almost all those bad days, I would like to sleep over, sometimes more than one day. If the wound is a bit deep, it will be sometimes 2 to 3 days. So I generally don't respond to something that I feel immediately. And generally, most of the time, even if I write a draft of an email when I'm upset, I put it into drafts and leave it there. Next 2 days, I don't touch it. And then I look at it, and if I still feel it is not, if it is not really working out, I actually send it to some of my friends and I'll say, what do you think? So they tone it down for you. So you should always be not reactive, and you should really kind of think your problems through.
I think, yeah, I kind of agree on it. Definitely.
I believe that if you realistically are reactive, and it happens to all of us at some point of time or other, what you would realize is it never plays out well.
Sir, so so today also, like most of our audience which joins, usually they are very young, maybe in UG or PG. So would you like to also give any tips to them that, okay, when you are in UG or PG, what should be your focus?
See, there are two aspects of it. One is our life, and the second is our professional life, right? So in terms of professional life, it is important to have focus. It is not how many hours you spend in a place, but it is how well you spend in a place, right? How effectively you use those hours. Focus, you have ability to troubleshoot your experiments if you're an experimental biologist. Right? Disappointments, how to get over the disappointments when they happen, and how to really start it all over when something just doesn't work out, or in worst case scenario, you're scooped, to keep yourselves still standing. It requires a lot of focus and lot of tenacity and lot of various aspects of personality. These things can be held in a personal space if you have a decent enough personal area, like you have good friends and you have good personal interactions with people. I mean, not necessarily only virtual, right? Somebody you can talk to kind of helps in getting over those disappointments. I believe that this is an aspect which was there for me in both ends all my life. I always had a bunch of people whom I can talk to. And there was, professionally, I was reasonably have this particular philosophy that, okay, what can worst happen? Let's have a relook at it. And then luckily I had a mentor who had a very similar kind of a of my lab come and tell me that something isn't working. I was like, don't tell me something is not working. Tell me what have you done. And when they tell, the second answer that you get is, no, I have done everything as per the protocol. Then I tell them, if you have done as per the protocol, then you're not going to be talking. You would not be talking to me. The fact that you're talking to me means something somewhere is wrong. So now you cannot troubleshoot if you listen to these two lines. You can only troubleshoot, and I tell them, don't look at a page, pen and a paper, close your eyes and tell me what exactly have you done. Because people say they have looked at a protocol and done it. I don't believe it. If they've done it, it should work. Right? So you need to have this particular, develop these skill sets. Troubleshooting is a very important skill set.
Yeah, completely. I agree on this. Sir, since you also mentioned about mentors, having right mentors in the life, so do you still have mentors?
Yeah, I mean, my
Do you,
PhD mentor, my postdoc mentor passed away because of cancer, and my other postdoc mentor has gone into something other than being a laboratory scientist. But my PhD mentor, I have a fantastic continuous relationship. In fact, we have had few collaborative publications, and I
Mhm.
continue to have very, I mean, I would say positive relationship with him. And I mean, I can call him anytime, and vice versa, and stuff like
Mm.
that. But you don't, they are not exactly the people you call and say, I have this problem anymore.
Yeah.
They are essentially, you respect them and you
Mm,
have this
mm,
particular
mm,
warm feeling for them, right?
mm,
And
mm, mm, mm.
leaving that part of it, it's not like I call him to say, I have this problem, can you solve it? Because I
Yeah,
am the
yeah.
person now, I should be solving it, right? So, but so, but mentors play a role at the initial years, and it is important to have good relations with them. And it's a two-way street. It's not. It's not that only one person is at fault when a relationship, if any kind of thing doesn't work out.
Yes, sir. Sir, your statement was really, really, really nice.
It's just two-way street.
Completely agreed.
Mm.
Sir, so apart from science, do you have any hobby that you like to pursue?
I generally play badminton, and I'm
Mhm,
pretty
mhm,
decent at it. I mean, I've been playing badminton since 2008, 9, and then in between, I even
mhm,
broke my ACL because of that. And then I got it fixed. I got into running, jogging actually.
mhm,
Almost 10 kilometers I used to run at a time. Then I slowly stopped that, and then now back to badminton. So every day
mhm,
morning, I actually go to play. Even today, for around 40 minutes, I play almost as much as possible. Not every day, at least 20 days a month, I play. But I play around
mhm,
40 minutes, and that obviously keeps me fit. And that particular feeling actually translates into a better day. So
mhm,
in
mhm,
between for
mhm,
a while, and because I couldn't manage my life well enough, I wasn't doing
mhm,
this playing part of it, or actually I wasn't doing any physical activity. I
mhm,
realized that
mhm,
comes into the other aspects of life. You don't feel very
mhm.
good, and that slowly gets into even your professional space. So physical activity of a certain amount, at least 30 to 40 minutes, helps me in keeping my more positive spirits.
Yeah,
I
yeah.
like
I
playing.
think
And I
sports.
used to play even as a child. I used to play carom, bit cricket.
Mhm. Yeah. So certainly the sports definitely help you to keep fit, even in the longer
Not,
run to
it's not the
work every
fitness.
day.
I'm not talking about the fitness, physical fitness. It's the mental fitness.
Mental fitness as well.
You
Yeah.
actually feel good. You
Mm-hmm.
feel very good after doing a 40-minute rigorous, exercise kind of a thing. When you actually go to the lab, you don't have this, you are more positive. That's for sure. I've seen both things. That's what I'm saying. You're positive, more positive.
Mm-hmm.
It helps to do some hobby, have some hobby.
Definitely, sir. Yeah, absolutely, sir. Sir, so can you shortly describe the biggest discovery that, or maybe the favorite discovery of your lab that you made in the past year?
So originally, no, in the last past year, but it's a journey. Originally, I used to do biochemistry, mostly looking at
Mm,
CCMB kinases, identifying targets, identifying phosphorylation sites. And after around 5 years, then I got into biology. For that, I started tinkering with the pathogen, right? And we
mm,
started looking into making genetic mutations in mycobacterium tuberculosis and
mm,
uh, uh, and other things. With mycobacterium tuberculosis, many manipulations, and slowly and steadily we started addressing questions much more deeply. And so some of my students have done a fabulous job of addressing fundamental questions in a very deep mechanistic detail. And so there are some fantastic papers that have come out and by some of these fantastic students who are in the lab. And so there is a lot. I mean, recently we are currently working on cell division and we have identified a new novel role for one of the proteins that is a cell division protein. Yeah, it's not that you do it. It is done by a student in my lab. Her name is Yogita. So she has done that. So there are always talented students who come up with exciting science, and that keeps you going, right? So, and I had some other fantastic students who have done fantastic work. Papers got published in good journals, and it's not easy to get into those journals sitting here. But nevertheless, we had one fundamental discovery where we showed that interaction of a ligand with extracellular domain of a kinase, okay, is not required for activation, but required for modulating the activation. Means without that interaction, it is higher activity, not lower activity. That's what it says. But if I abrogate the interaction, it is more active and it loses its localization. So that is something we have shown. At a later point of time, we identified a transcription factor, not transcription. Yeah, we identified a transcription factor which has one role and one role alone, which controls cysteine metabolism. And that to an alternate cysteine metabolism, and that is required for the pathogen to survive in the host. It is not important normally, but when you go into the host, when the stress comes in, oxidative stress comes in, cysteine as an amino acid becomes an important one, because cysteine then feeds into antioxidants like mycothiol and other things. So these antioxidants are not produced enough in the absence of that particular activation by the transcription factor. This was a paper that we published a few years ago, fantastic work. And there is another girl who has done find novel mutations by doing a GWAS study in specific novel mutations in DNA repair enzymes. So she has looked at multi-drug resistant pathogens, and there were certain novel mutations were identified in the DNA repair enzymes. And then we went back and validated that this mutation abrogates the activity of the DNA repair enzyme, and it provides some survival advantage you put any stress, including antibiotics. So this we have shown, and that's another good piece of data. And right now I was saying that we are looking at cell division, and we have shown that one protein, specifically a protein named BAC31, has two distinct domains. One domain is required for interactions with the lipids at the membrane, and the other domain is required for actual interactions with the proteins. This was done very systematically. So that's not published yet. But these are the kind of some of these interesting findings over a period of last 5 years is what I talked about. And there are many more interesting findings previously. Every 2 years, you have something that you are excited very heavily about, right? And then you actually publish that paper, and you ultimately talk about it in every meeting possible. It keeps you going. So you always have to, that keeps on changing as the science progresses. So it's something that keeps you active. So I have had some interesting findings, which I talk about it all the time. Right now, we are involved in sequencing 31,000 clinical strains of tuberculosis in India. And we are one of the labs, and we are doing 13,000 in CCMB. Already, we have sequenced around 2,500 in CCMB. And this is my lab directly, not some other lab in the. So I have two or three people who are working on it, and we have already sequenced 2,500, and we have to achieve 13,000 from our end by next year and this year, the 2025 end. And I think we can do it. And if we do it, then we will get to know what kind of strains are there in India, and what kind of persistent patterns are seen in India, and stuff like that. So there will be, this is a huge collaborative project which involves ICGB, NIBMG, NII, report cohorts, a hugely collaborative project which involves people with very different set of skills and across the board, clinicians to people who are involved in clinical, because they have to get the strains, and then people who are isolating DNA, making repositories, and we are doing sequencing, data analysis. It is something, these are the kind of projects that go abroad, that go on and in US and in Europe. And I think we will be the first ones to really attempt problem at this scale. So many people just doing parts of a big problem. So that is exciting for me.
Definitely, sir. So how do you see, sir, India as an growing, I would say a blooming flower, now, or
Yeah,
maybe
sure,
which
sure.
has already bloomed?
No,
How
there
do
are
you see this going ahead as a
lots of
director?
lots of institutes have come compared to where when I was looking for a job, and a
Mm-hmm,
lot more
mm-hmm,
when I was a student compared to when I was a student. So yes, the opportunities are more, but and a lot more talented faculty or talented people in various places, and you interact with them, and their expectations are high. Sometimes the country actually doesn't. I mean, like we have rules and all which are a little more difficult to deal with for them. But I believe every country, every place has its own set of challenges, and we now have a lot of people who are working well. And so I believe there is a huge opportunity to do better. But I think the whole focus is shifting towards societally beneficial science also. So there is an emphasis on, where very good you're doing fundamental discoveries. What else, right? That aspect is coming. So I have done a lot of fundamental work too. Now I'm looking into what way I can use some of my skill sets for doing a clinical work of the kind I said, being a cog in the wheel, than the wheel, right? So,
yeah.
right. So it's exciting. Opportunities are there. People have to have a mindset where they have to realize that in addition to doing what they're doing, they should at least put certain percentage of the effort to do something what the
Mm-hmm,
country needs you to do or wants you to do. I mean, not
mm-hmm,
necessarily a defined project, but something that you can
mm-hmm,
think in what way your skills can help in doing that. And if you can,
mm-hmm.
what is that, marry these two particular aspirations, which is personal motivated research with something which will be beneficial in a larger scale, most probably we are going to do much better going forward.
I see. I see, sir. Sir, so like I always ask this one question to everyone, and I would love to ask you this question to you is today, like which is the one discovery in the scientific world till now that you see as your something like a discovery that you really give importance. One of the PI, I would just give you a rough idea was to answer this question. One of the PI who came who really loved the answer was the discovery of HCl, who said that when there was a discovery that yeah there are H plus ions and Cl minus ions for the first time, and from where the chemistry and the biochemistry and biology started. So as per you,
Fundamentally, this question can be two, three stages. Fundamentally, if you go
uh-huh,
back to 1950s, the structure in
mm-hmm,
itself is fascinating. And then
mm-hmm,
came
mm-hmm,
the whole Quran, how they broke the what codes for what. That's like
mm-hmm,
amazing discovery. Right now, a
mm-hmm,
little later in the life, how the sequencing has evolved is changed the way our
fantastic.
complete way we look at it, biology. And from the
Mm,
context of major findings, I believe it is the regulatory findings, how it is CRISPR and mRNAs. I mean,
mm,
literally
mm,
think of this landscape.
mm,
In 2000, people did not know, right?
mhm,
And
mhm.
today, you talk about doing gene therapy with CRISPR,
Yeah,
right?
definitely.
And it's obviously a very, very highly influential finding, and that is going to change both. It's a healthcare sector. Now, think so. For me, it is not one unique answer. There are some major discoveries that have changed the way biology actually happens.
Mm, mm, mm. I completely agree on you. And also, like I forgot to ask you one question, which was very impromptu in my mind. So when I was traveling, I was talking to a computer scientist, and there what he talked was that even in biology, but I would like to have that answer from you. He said that the way that the computer science has changed completely to AI and algorithms, which was not when they studied the computer science. So how do you see this transition in
Biology,
biology or
huge,
biochemistry?
huge, huge. Just think of it. In 1990, Human Genome Project was kind of announced around 1890. And what is the belief that you sequence the genome, you solved everything. right? And the technologically, it was so difficult a task. It was $1 billion kind of work, right? And by the time it got solved with various technological developments and computational developments, it became, it took 98, 97, 98, I think that's when it came out. Now, you sequence the whole human genome in less than 24 hours.
Yeah.
Then you then talk about, what is that? You're talking about RNA sequencing. You're talking about epigenomes. You're talking about various levels in the human genome. This technological transformation from just doing a 400 base pair read on a gel to literally doing a whole human genome in 24 hours is by far the biggest jump I have seen in last 35 years. Right? I mean, this single-handedly transformed the way science works, next generation sequencing, and the technological evolution that's happening with it on a daily basis, or literally every year things become obsolete. I mean, the speed at which things are happening in biology is both exciting and scary. Right? I mean, what was today scary because sometimes you really can't keep up with what's happening around you. And that's what I'm saying, you can easily become obsolete in this field in 5 years. Right? If you continue to do science the way you were doing 5 years ago, you are obsolete today. That is the reality. So this is one of the biggest transforming things. If I want to, let's say 1990 to 2024, 400 base pairs with great difficulty to whole human genome in 24 hours. That's the transition. And
Completely, absolutely.
And other way, floppy drive of 1.4 MB, floppy drive of 1.4 MB to a pen drive of 64 gigabytes. Tape recorders.
Absolutely.
Where tape recorders do even exist? Not there. It's like an item that is out of everybody's memory. Your generation doesn't know. The best you can think of is CD. Even that's gone. Everything is digital.
Yeah.
Now it's cloud.
Yeah.
Right?
Yeah. Everything is on cloud.
Yeah.
You upload everything on iCloud or maybe on...
I mean, you are sitting in Germany, right?
Germany, yes.
You're in Germany, I'm in India. This conversation, few years ago, out of question.
Definitely. Just before COVID as well.
Yeah.
Nobody thought.
Right? So amazing changes. Sometimes I'm saying, no, for me and my scientific career, maybe another 10 years. But for you guys to keep yourselves at that level, it's a constant battle. And there's, something that you need to understand, that you can never stop learning.
Definitely. Sir, have you imagined after maybe 20 more years when you would retire, or maybe soon, but after 20 years when you would already be out of maybe institutes? I mean, have you imagined the science or the biology that could move?
I think it'll be exciting, but it'll be very different. I'm very, very sure of it. Personalized medicine is going to become norm. It's going to become norm. I would say another 5 to 10 years, upper limit. Everything will be personalized medicine. And gene therapy will be part and parcel of our skill sets for, treating cancer. I mean, CAR T is going to become better. And gene therapy would be better for even most probably resolving genetic disorders. So genetic disorders today are challenging, maybe curable. So it's going to be huge. I mean, of course, money may be something I really can't talk about, but from the medical advancements and the scientific advances, I believe all these things will be feasible in about 10 years. So that's what I feel where we are heading, and all the more. And one of the things is, you already know you can put a synthetic gene and synthetic whole genome of something like E. coli. So how far are you from doing that for humans?
Definitely.
So there is so much. I mean, of course, ethical constraints and ethical problems are always there, but there are things that are going to be different, and I believe this is the direction in which we are going. And how far we can go, it's sometimes very difficult to imagine. And there was a time Crick actually stated that all the introns are junk DNA, and no none less than Crick of Watson Crick, Francis Crick. There is an
Definitely.
article which says junk DNA. I mean, article. So beliefs are so, what's true today is not true tomorrow.
Absolutely, sir.
So,
Sir, so I think today's event was really, really nice. I really loved the interaction, the formal one, and also the informal way that you had a conversation. I really loved it, and I really look forward to see you when I am in India once.
looking forward to be.
Sir, so
Hmm.
we have some questions on YouTube Live as well from the participants. I would also like to take very few from them. I really feel sorry that I couldn't take all the questions due to the time constraints. But one question which I would also like to ask you and also the audience was that when you go for the interviewing the students, what qualities or skill set do you look for in a student if you want to uh,
It's the
maybe
universal
get.
answer. It's not a student. It is whether it is a student, faculty, anybody whom I interact with. The thing that it generally look for is that there has to be a certain level of positive energy.
Mm-hmm,
You
mm-hmm.
can't come into the room looking literally why am I here, right? I need that positive energy. And the second thing that I look for in a student is ability to think out of the box. Means if you are going to give me textbook answers, what's the big deal? Right? If
Yeah.
I give you a theoretical situation, can you work it out? At least make a proper efforts to work it out. And at least even if you come 30%, 40%, the fact that you're thinking is a very positive, right? So these are the two things that I believe is body language. You cannot be like walking in dead, and then you need to be able to think rather than shut your mind and say, this I do not know. So always be that person who says, I do not have an answer, but I can work it out. Or at least I'll give it a shot. I'll try. That's a positive answer.
I think I agree on this. Yeah. So, sir, also one more question, a bit towards a technical side, toward molecular biology or omics. A student is asking that can you identify the environmental factors through multi-omics screening that may affect based upon the climate change or
Yeah, you can.
maybe.
You can. Most of the time, and you may have seen few papers that are coming here and there, epigenomics is where the changes are going to be. So environment dictates changes in the epigenomics of your this thing. It is not the genomics anymore, it's the epigenomics. Epigenomics directly controls transcription, which essentially means the expression is going to be the protein content is determined by the epigenomics. So epigenomics are environmentally driven. So suppose you are in a place where you don't get as much food versus a place where you are well fed. That's an easy control to actually look at it and look at their epigenetics. You will find differences. And recently, you've seen that there was a paper which talked about intermittent fasting, or there was a paper which talked about people who are actually overweight and lose weight, and it is very easy for them to get it back because their epigenetics don't change drastically, even though the lost weight. Epigenetics has long memory. So yes, you can correlate environment, but it is not at a genetic level. Genetic level, you can correlate it if there are mutations, but if there are no mutations, you should correlate it with the epigenetic
Yeah, completely agreed on this. So, sir, I would like to say thanks to all our audience today, and also especially to you for joining the event on Sunday morning, taking out time on Sunday because majority of the PIs are equally busy, and you gave us time. I'm really thankful towards you. So I would like to request Soumya Mishra. Soumya is doing currently PhD in Bark, and I would like to request him to please deliver vote of thanks uh, as part of the today's event. So
Thank you.
Soumya, please.
Yeah, really. Thank you, Arya, uh, for your giving the opportunity to me for providing for help means for giving this opportunity. Thank Dr. Vinay Kumar Nandicoori for giving us such a great, insightful talk about his journey and what and his contributions in the society. And it's really, I am really thankful to you, sir. And fortunately, it is my pleasure that I have also got the exposure of working in the Mycobacterium tuberculosis lab in the at ISC at Amit Singh's
Oh,
laboratory in
okay.
the
A good friend of mine. Yes.
yes, sir. So and now currently I am working on exosomes as a radio protectors at Baba Atomic Research Center. So with this, I really thank you, sir, and I hope we can do some offline event with you if possible.
Sure.
So that yes, sir.
I
And
always like interacting with the students because it is something where you actually, I mean, it's not necessarily that I have the best message, but it is the best possible way to directly communicate and tell them your apprehension, not yours. You all pass through that
yes,
alone.
sir. Really,
Certainly.
thank you, sir. Thank you so much, sir. And
Thank you.
yeah,
Thank you.
so with this, I would like to thank all of our audience who joined this event, and we will be pasting our feedback form for the e-certificate in our live chat box so that you can fill it and get your e-certificate with that. And I'm really thankful to everyone. And please do like, share, and subscribe our channel, the SciComm Synapse. Till then, think, share, and grow.
Thank you.
Thank you
Thank
so
you
much.